So for this experiment, first we're gonna culture 293 T cells, and then for the first two are gonna be UV damaged, and then these are gonna be just normal. And then in this, we're just gonna use the cell. But for the second one, we're gonna buy siRNA against P53, and we're gonna transfect that P53 siRNA. And then this one's gonna be our cell control. That's not UV damaged. That's number 3. And for number 4, a cell that's not UV damaged, and then we're gonna transfect P53 siRNA. So after this, what are we gonna do? We're gonna transfect the P53, like I said. These guys are just gonna be cells, and then or we can do like a control siRNA. It doesn't matter really. After that, we're gonna wait 24 hours after our transfection. After we wait 24 hours, we're gonna do qPCR to measure the mRNA for P53, and then we're gonna do Western blot. While we do this, we can also transfect a luciferase using the GAAD45 promoter and luciferase LRG. So why are we doing that? Because we're trying to check if decreasing the amount of P53 will affect the GAAD45 promoter activity, basically. So what do we expect from our qPCR? 1, 2, 3, 4. So first and foremost, P53, we only have P53 protein when our body is damaged. So in the minus UV, we shouldn't expect any P53 protein, or in this case, well, mRNA, because we're looking for mRNA, P53 mRNA. So we should expect almost nothing. And then for our cell that's UV damaged, we should expect a lot of P53. But for siRNA, because we expect the siRNA to basically reduce the amount of P53, we should expect less amount. And then our Western blot, we should expect large band for number 1, just because there's P53 there. But for number 2, because of the siRNA used, we should expect small amount of P53, and the same for the cells that are not UV damaged. So now the last thing we're going to do is the luciferase assay. So in the luciferase assay will only work when there's a GAAD45 promoter, and GAAD45 promoter, we expect it to have activity when there is the P53. So we should expect in our luciferase assay for the first one, we should have a light present like this, and then for the rest, it's going to be something like this, because there's no P53. We shouldn't expect the GAAD45 promoter to be active. So this would be our results basically, and then this will tell us that decreasing P53 basically also decreases the promoter activity for GAAD45. Thank you.